Okay, thanks, Anoop. Can everyone see my screen now?
We see a
Yes.
dock.
Yeah, see a doc. That's perfect. Yeah. Great. This is Fang Ming from MooCamel Lab. From UCSD, and today I don't have a formal presentation. I just want to do a quick demo um, of the tools we built from the MooCamel Lab. Um, about showing the multimodal molecular profiles for brain cell types. Um, so we are fortunate to have different data generation groups providing different data sets. And we gathered all those different data sets, including DNA methylation from Joe Acker's lab, which Han Qing has already presented his version, and open chromatin quantified by single cell ATAC-seq provided by Ren's lab from UCSD, and also gene expression data from Allen Brain Institute and McCoskell lab from the Broad. So we gathered all those data types together and did integrated analysis. I won't mention how we did it, but it's in the associated preprint, and I think Han Qing's and Young's, and it's highly related to Han Qing's and Young's work. So I'm going to do a quick demo of the results and the tools we built to visualize the multimodal profiles of brain cell types we found. And besides myself, today we also have Ethan Amand, who's my lab mate, who's also here, and he might be also able to answer questions if you might have, either in the chat or later after the quick demo. Yeah, so let me get started. Oh, before I get started, let me just share this link to you guys so you can all access it and play with it. So my goal today is to give everyone a hands-on experience of playing with the tools we have, and hopefully helping you overcoming the initial hurdle of using the tools, and you might later explore it more and find it useful, hopefully, in your own research. So I have two browsers to show you today. One is a genome browser, um, and the other is a cell portal. Oh, so let's first just go to the genome browser. Um, and if you click that link in the doc, or if you go to brainome.ucsd.edu/BICCNMOP, it'll direct you there. And here comes the default view. It takes a few seconds to load up the tracks. So this is a genome browser or NOJ browser. The basis of this browser is initially developed by several people from Joe Acker's lab. So we took that basis and configured it to show it for our purpose. So just to take a look at this default view, you see a bunch of different things. And the top tracks, if you're familiar with genome browser, the top tracks is showing you the annotated gene models, gene annotation of the mouse genome, MM10. So then each track down here shows you some cell type profiles, cell type specific profiles of the molecular profiles across the genome. For example, if you focus on the first track, it says MCG, LAM5, something, something. MCG means it's a DNA methylation in the CG context. So DNA methylation occurs across the genome. So the height of each tick shows you the methylation level of that, the methylation level at that site of that particular cell type, LAM5, PDLM5, something, something. For example, if we go to a gene, palvobumin. Oh, by the way, you can search gene from here, and it'll direct you to that gene. here's a parvalbumin, and in that gene, you see that the DNA methylation. So here, if you look across multiple different tracks, you'll see that the height of the ticks varies across cell types. That's because the DNA methylation levels are different across different cell types. And it's particularly low in this track. This track is marked PVLB, so it's PV cells from the mouse brain, and it has specific cell type specific low DNA methylation at this gene parvalbumin, but not in other cell types. And interestingly, although it's known for a few years now, but still, what I think is still remarkable is you can see a corresponding ataxic peaks down here in the same cell type, PVLB, reeling cap5 C5 _1. You see a corresponding ataxic peak suggesting regions of open chromatin only in that cell type, but not in others. So that's the general flavor we want to give you for this type of browser. You have multiple different cell types, and you have corresponding multimodal profiles for each of those cell types, and you can jump back and forth in to any genomic location you like and explore their profiles. So there are many different settings you can configure besides the default view we provide here. For example, if you go to the top menu, the menus here, you can choose which cells to show. By cells, we mean cell type, which cell types you want to show. If we click inhibitory neurons, and the level of the cluster depth means the resolution of cell type you want to look at. You could look at fine level resolution like cell type, or broad level cell types, cell classes, or cell classes. Let me choose just cell classes for now, and you can select the data modalities. So apart from the DNA methylation, MCG, and open chromatin attack and the enhancers, we can also select, for example, RNA, single nucleus RNA, and you can group tracks by data modality or by cell type. Let me just group it by modality and click update or reload browser. It'll update to the configuration you selected. Now it changes because I selected an extra data modality, single nucleus RNA. So this modality also shows up. Unsurprisingly, you see gene expression. So the third type of data modality shows you gene expression. The gene expression of parvalbumin also shows up only in that cell type, but not in others, if that's clear. Also, if you have question, please feel free to leave it in the chat or interrupt me. Another thing we could do, for example, is to group the tracks. You can select the orders you want to at which you want to display the tracks. For example, if I want to group the tracks by cell type instead of by data modalities, like it does now, I can select this. I can select group by cell type and reload again. Now it regroups the tracks. It still shows the same track, but regroup the orders such that each cell type, the different modalities of the same cell type, are now together. Now it's more clear. Even only in one cell type, the parvalbumin cell type, you see low DNA methylation and open chromatin and gene expression only in that cell type, but not so much in others. So hopefully this gives you a flavor of what's in this browser. And of course, you don't have to focus in the gene body region. We could go to another example, for example, tag 1. Okay, and so you could go to, besides gene body region, you could go to non-coding regions of the genome, and the informations, the profiles are, the epigenomic profiles are still there, but the gene expression profile is only limited to the gene. So, for example, this is not a big gene if you count from the start side from the beginning to the end. It lasts around 10 kb, and so you can click zoom in and zoom out from here to show different to show the profiles at different resolutions. One interesting thing we found is, although we know that this gene seems to have a cell type specific, strong cell type specific signal at this parvalbumin cell type, and in this gene, we also found some more subtle but still strongly cell type specific signal in the non-coding regions upstream of this gene. I'm pointing you to a little ataxic peak here that's specific to this cell type, and you have a corresponding low DNA methylation as well. And that suggests a potential enhancer region likely regulates the expression levels of gene. So you can go on and on into this non-coding regions all day to see the vast profiles of these cell types. So besides the default views and a few configuration I just shown now, I guess you can also look left to this configuration or track selection panels. For example, if you want to move this track up, you can click the corresponding track here and drag it to the top. Now it goes to the top. You can do it however you like. Yeah. And goes like this. And and you can also adjust the scales of the tracks. For example, if you found this particular track not obvious enough, you can change its scale. Now it's scaled at 0.6. If I tune this larger to 1, you see it's growing. And if I change it to 2, it grows even bigger. What else? Oh, you can also, there's many details you can explore. For example, here you can tune up or down the scales globally, or the height of the track up and down like this. And you can jump to a particular location of the genome. This is now chromosome 6. We can jump to chromosome 10, for example. Another useful thing is you can save this configuration you created and share it to anyone else, or save it for further explore sometime later by yourself. The way you do it is, after you click reload browser and make sure everything is updated as you like, you just copy this long URL. This URL registered all the configuration you made as you explore. And if you copy that URL and enter it sometime later, it'll show up, it'll return to the previous configuration you have set. For example, if I open incognito view, so it doesn't remember what I did before, but still, if you have saved this URL, this link, it'll guide you back to where you were, essentially. So that's about what we have for this browser. And besides this browser, we have also included the external resources like BICCN, the Nemo page, Nemo Analytics, and an anatomy browser developed by Hongwei Dong's lab from UCLA, shows the which shows the anatomy of the mouse brain. And so this data set is entirely from the mouse MOP, but going further, we might explore different brain regions, and it's useful to have a corresponding anatomy browser. And also single cell portal from the Broad. I think many of these tools have been presented today already. And one last thing I want to show you is a cell browser showing the same data essentially, but gives you a different view, the t-SNE and UMAP view you have seen today. So this is a corresponding UMAP view showing the distribution of single cell and their relationship in the t-SNE colored by cell types, or colored by the gene feature. Here it's showing you normalized gene body methylation level. You can click up left here to configure it to different data modalities. So now I'm switching to ATAC-seq panels, and you could also switch to ATAC to RNA as well. Because all the cells are in the integrated embeddings, so the embedding doesn't change much. The cells are grouped by cell types instead of data modalities, but the gene features are different. You can view from gene body methylation to gene body open chromatin level or gene expression level. So that's about the tools I want to present. If you have questions, I can take it now. Yeah, otherwise, yeah, let me just pause it here. And any questions? One fun thing to do is to have everyone name your favorite gene, and we can just all look at it together. Okay. Yeah, if you have further questions, feel free to contact us and to RFX3.
I mean, somebody put in a gene there.
I see. Yeah. Okay. Let's go RFX3. Okay. Here's the full gene body of this gene, because I'm now only showing you the inhibitory cells. Let's include all cell types and reload. Question, can you also go over the different tracks in the scale tracks dropdown? Yes, you can also go over different tracks in the scale track. I'm not sure if if I understand the question. Do you mean if I can go over the tracks from the left, from what I'm pointing to? Oh, different options in the drop down. Yes, definitely. So here are the different options. Let me deselect all first. Then nothing is selected, only the gene annotation is here. So MCG is the DNA methylation level at CG context, at CG dinucleotide. So MCG looks like this. The green ticks goes on like this. MCAC is DNA methylation at CAC trinucleotide context. It's the C followed by an A and the N followed by an C. So that shows you the non-CG methylation profile. Because in DNA methylation, there are two features, distinct features in general: CG methylation and non-CG methylation. The CAC represents a typical non-CG methylation level. So those are the blue tracks here, and you see they're very different. ATAC shows you the chromatin accessibility, and they are marked by one limit of this browser is as you as you select too many tracks, it gets slower and slower. But let me focus back on a few smaller sets of cell type instead. Okay, now the blue tracks shows you the chromatin accessibility. You see this blue peaks concentrated around the promoter of this gene. That's chromatin accessibility. Single cell RNA-seq and single nuclear RNA-seq shows you gene expression. Single nuclear means that they collect the RNAs from the nucleus only, but cell means you collected RNA from both cytoplasm and the nuclei. So that's shown in this red, dark and light red colored tracks. I'm not sure why it show up in non-coding region for this particular gene, but let's go to a different gene, for example, GAT2, which is an inhibitory neuron marker. You see the gene is expressed in GAT2. Enhancers are putative enhancers generated by an algorithm called Reptile that's developed by a graduate student initially from Juaker's lab, and the analysis is run by Han Qing. So we just took the result, and they predict the putative enhancers across genome. So these little black sections indicate candidate enhancer regions that the algorithm think the algorithm think might be an enhancer region, cell type specific enhancer region. And you can see the reason why it thinks it might be an enhancer is because in those regions you see a small peak of open chromatin and low DNA methylation level, and it tends to be cell type specific. So that's all the available tracks you have. Does that answer the question? Or okay,
there was
thanks,
a request
Andrew.
for one more gene, I think, in the mice. If you want to look at that, it's from Michelle Engel, around 353s. Single CF.
Oh, single
Repaste
CF,
it.
single CF for mice. Let me copy that over. Single C. Okay, that's this gene. It looks like it's not expressed in any of the cell types shown here, shown the MCG derived neurons. Let's switch back to all cell type maybe, and select the cell depths. Maybe show fewer tracks. Okay, I think while it's loading, I think we're right on time. And thanks everyone. If you if you're patient enough, we can wait until it's loaded. Otherwise, my presentation is done. Okay, now it shows you all the cell types at this gene location, single C. I'm not sure about single CF. Does it? Is it a different gene? It looks like it's not expressed in any of the cell types here.
Okay.
All right, thanks.
Well, Fangming is still, are you done, Fangming, or do you want to wait a little bit longer?
I'm done.
All right, thank you very much. I'd like to sort of take this opportunity for thanking all the people who stayed on with us. I know it was a bit of a conflict with the presidential inauguration and us organizing this workshop, but you persevered and stayed with us. Thank you very much. I'd also like to take this opportunity to thank all the presenters for all the work they've done in sort of bringing all this together. And we will be sending out a short survey fairly soon. So if people could sort of answer those, that'd be great, because we'd like to give feedback to the presenters as well as the organizing committee of the workshop itself. And I just want to remind people that this week, the whole focus was on essentially GUI-based tools where you can do interaction. And next week, we'll be shifting gear towards tools that are more designed for people who want to do primary analysis, although snuck in there is a single cell portal, which is kind of similar to what you saw today, but there'll be a little bit of presentation on single cell portal next week as well. So see you all next week. If you don't have any other questions, we'll sort of sign off. And if you do have questions, you can send us an email or reach out to some of the organizers.